Hello, am I audible? Please let me know in the chat box if I am audible or not, and also please let me know if you can see the video. Okay, thank you for confirming. Can you let me know if the presentation is visible to you? Let me know if the PowerPoint presentation is visible to you or not. Okay, thank you for confirming. Let us wait till it's 6 p.m. Then I will start today's session. Okay. Meanwhile, if you have any doubt or any query regarding the previous classes, you can let me know. Otherwise, let us wait till it is 6 p.m. Then we will start today's session. Okay. We will wait 2 to 3 minutes more. Okay. Once it's 6 p.m., then I will start today's session. Meanwhile, if you have any doubt or any query regarding our previous class, you can let me know now. Okay. So good evening to the students who have joined. I hope my voice is audible, and I also hope the video and the presentation is visible to you. If not, please let me know through the chat box. Okay. We will wait till it's 6 p.m. Then we will start today's session. Meanwhile, if you have any doubt or any query regarding our previous class, you can let me know now, or you can let me know later also if you have any doubt or any query. We will be starting our session soon, so please wait till then. So I think it's 6 p.m. and I think I will be starting the session. Okay, let more students join. But let me start with the introduction. So very good evening to the students who have joined. My name is Mousumi and I'm a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL SWAM course of Organic Chemistry in Biology and Drug Development. And today it is our week 10 live session. So in every live session, what we do is like we discuss about that particular week's course content via 20 MCQ questions. Okay, these questions will help you to understand the course better, and also it will help you to answer your weekly assignments. Okay, so before starting, let me clarify a few things. Okay, so the thing is like there will be a problem solving session every week. Okay, and these sessions are scheduled on every Friday from 8 to 6 p.m. These sessions are live on Google Meet platform, and this is the link for the Google Meet class for our entire session. The link will be same. So the important point is if any network disruption happens from my side due to internet connection or due to power cut issue, please do not leave the session immediately. I will join as soon as possible. During the session, the students can post their queries and doubt through the chat box, because I am recording the session which I will be sharing with you after the class. Okay, this recording will help you later also, so that I can record the session without any issue. I have muted your microphones and cameras, but the chat box is always open. So you can let me know every query, anything, doubt, everything. You can let me know via chat box. The chat box not only we will use for the answer for you to ask the queries and everything. Also, we will use for answering during our question answer session. Okay, because I will show you the MCQ questions which will have 4 different options, and after I show you a particular question, I will wait for 2 to 3 minutes, and you have to identify A, B, C, or D, which of the following will be the correct answer for that particular question, and you have to let me know in this chat box. Okay. This presentation and class recording will be shared with you. Okay, so if you have any issue with your internet or with your connection, the call is open. You can rejoin at any time. Okay, so no worries with that. And since I'm recording the session and it will be shared with you, so you can check the part what you have mixed later also. So where you will find this presentation recording and this presentation. So you have to. I have shown you earlier times also, but let me show you one more time. So you have to go to this NPTEL SWAM portal, where is the on the course page. Okay, you will find here the weekly assignments are posted along with the lectures are posted along with your quizzes and feedback forms and everything. So you will find there is a left panel which contains a problem solving session tab. If you click on this particular problem, you will be redirected to the problem solving session recording. Okay, so it will take you to a page where you will find every YouTube link for the every class. Okay, whatever we have done so far, we have till now we have covered 9 weeks. Okay, and today it is a week 10 live session, so that 9 video and all the presentations you will find in the by clicking to this page. You will find the YouTube links also. You will find a Google Drive link which contains all the presentations. You can download the presentation and you can go read the presentations from there. Okay. So today it is our week 10 live session. So as I have mentioned earlier also, so in our last class, in our week 9 live session, we have basically discussed about many things regarding your drug discovery, right? So we have discussed various steps of drug discovery, how the very phases of the drug discovery and clinical trial happens, what is the preclinical studies. We have discussed about the pro-drug therapy, what is the logic behind it, and we have discussed about various neurotransmitters, and also we have discussed about the renin angiotensin system and antihypertensive drugs. Okay, so these were the topics which were discussed in our previous class. So if you have any doubt regarding the previous class, you can let me know now. Okay, also you can let me know via email. I will share my email ID at the end of the session. Also, you can post your doubts or your queries in the discussion forum. Whatever doubt or query you have during our problem solving session, you can post it in the discussion forum of the organic chemistry and biology and drug development. There also I will answer your queries if you have anything regarding my class. Okay, so as I have mentioned earlier also, today it is our week 10 live session. So first let us see what are the topics which is in our week 10 course content. So let me just make it full screen. Yeah, so our week 10 course content has these topics. We have antimicrobial drugs. We have to discuss about the chemistry of penicillins. We will discuss about the resistance to beta-lactam antibiotics. We will discuss about the mechanistic studies of beta-lactamase and non-beta-lactam antibiotics. Also, we will discuss about the mechanistic enzymology of isopenicillin N synthase. Okay, so these are the topics of our week 10 course content. I hope the presentation is visible. If not, you can let me know now. So let us go to the first question of today's session. So once I show you a particular question, I will wait for 2 to 3 minutes. Then I will check the chat box. A, B, C or D, 4 different options will be there. And whatever you think is the correct answer, you can let me know through the chat box. Okay. There is no marking system. So even if your answer is wrong, it does not matter. But answering during the question, during the session is very important. Otherwise, I won't understand that if you are following the class or not. Okay. Now let us go to the first question of today's session. So the first question is of today's session says, which of the following statements is not true for bacteriostatic agents? Your options are: number A, the agents stop the growth of the bacteria. Option number B, the bacterial population remains same. Option number C, the agents destroy the bacteria. Option number D, the agents inhibit processes like replication, translation, and transcription. Which of the following statements is not true for the bacteriostatic agents? Let me know in the chat box A, B, C or D which of the following statement you think is not true regarding bacteriostatic agents. I have told you earlier also many times that whenever there is a true, false or correct, incorrect type of question answer, please read the options and the question very carefully because most of the students they make mistake during this kind of questions. Okay. Let me wait for 2 to 3 minutes, then I will check the chat box. Which of the following statements is not true for bacteriostatic agents? Your options are number A, the agents stop the growth of the bacteria. Number B, the bacterial population remains same. Number C, the agents destroy the bacteria. Number D, the agents inhibit the processes like replication, translation and transcription. Which is not true regarding bacteriostatic agents? Which of the following statements is not true for bacteriostatic agents? Number A, the agents stop the growth of the bacteria. Number B, the bacterial population remains the same. Number C, the agents destroy the bacteria. Number D, the agents inhibit the processes like replication, translation, and transcription. Okay, I think I will check the chat box now. Let me see if there is any answer for problem number 1. Okay, so I got 2 answers. Both are saying that number B is the correct answer. Okay, now let me check if number B is the correct answer or not. So the correct answer means the answer which is not true regarding bacteriostatic agents will be, it is number C. The agents destroy the bacteria. This statement is incorrect regarding bacteriostatic agents. Now let me tell you why it is so. So what is the definition of the bacteriostatic? So there are two type of agents. One is called the bacteriostatic, okay, and another is called the bactericidal. So what is the bacteriostatic? So these are the antimicrobial agents that reversibly inhibit the growth of the bacteria, okay, like tetracyclines, chloramphenicol. These are the examples of the bacteriostatics. And we have bactericidal. Bactericidal agents are those antibiotic agents which with an irreversible lethal action on the bacteria are known as the bacteria like penicillin and isoniazid. So what is the difference? So bacteriostatic agents, they actually inhibit the growth of the bacteria, means whatever bacteria is present, they will not grow anymore. Okay, they will inhibit the growth. The number of the bacteria, the population will remain same, but it will stop the growth of the bacteria. But the bactericidal are those compounds which will kill the bacteria, means in the case when we are administrating a bactericidal agent, it will decrease the bacterial population. Okay, so that is why the statement number C is false, which says the agents destroy the bacteria. No, bacteriostatic agents do not destroy the bacteria. They only inhibit the growth or stop the growth of the bacteria. It is the bactericidal agents that basically destroy the bacteria. So what are the differences between the bacteriostatic and the bactericidal? So bactericidal refers to antibiotics that kill the bacteria and the action is irreversible, whereas bacteriostatic refers to the antibiotics that prevent the growth of the bacteria and these are the reversible agent. The action is reversible. irreversible. The bactericidal inhibit the cell wall formation of the bacteria, and the bacteriostatic they inhibit the DNA replication and protein synthesis of the bacteria. Bactericidal do not work with the immune system of the host, and bacteriostatic they work with the immune system of the host to prevent the growth and the reproduction of the bacteria. MBC refers to the concentration of the drug required to kill 99.99% of bacterial population, whereas MIC is the minimum drug concentration which inhibits the bacterial growth. This MBC is required for the bactericidal agents, and the MIC value is basically checked for the bacteriostatic agent. Examples of the bacteria include beta-lactam antibiotic, cephalosporin, and vancomycin are for bactericidal, and the example of bacteriostatic are the tetracycline. We have streptomycin, chloramphenicol, and sulfonamides. Okay. So this was all about your bacteriostatic and bactericidal agents. So do you have you understood this thing? So is there any doubt regarding the first problem? Why option number C is correct answer? Why not B? Because the option number B, it says that bacterial population remains the same. This is true. Okay, because bacteriostatic, they are not killing the bacteria. That is why there is no point of the decreasing of the bacterial population. Only their growth is stopped. Okay, so there will not be a decrease in the number of the population. Rather, it will remain same. Okay. So that is why the problem, and the B is not correct answer. Okay, the correct answer is number C. So if there is no doubt, let me go to the next problem. So problem number 2, it says that mechanism of action of penicillin. Okay, sorry. So problem number 2 says the mechanism of action of penicillin involves the inhibition of a particular enzyme in the bacterial cell wall. Identify it. Your options are number A, carboxypeptidase, number B, transpeptidase, number C, glycosylase, and number D, phosphatase. The mechanism of action of penicillin involves inhibition of a particular enzyme in the bacterial cell wall. Identify the particular enzyme. Your options are number A, carboxypeptidase, number B, transpeptidase, number C, glycosylase, and number D, phosphatase. What is the particular enzyme that is involved in the mechanism of action of penicillin? Again, I will wait to 1 or 2 minutes, then I will check the chat box. Meanwhile, let me know A, B, C, or D, which of the following you think is the correct answer. The mechanism of action of penicillin involves the inhibition of a particular enzyme in the bacterial cell wall. Identify the enzyme. Carboxypeptidase, transpeptidase, glycosylase, or phosphatase. Let me wait for some time, then I will check the chat box. So now let me check the chat box. Let me see if there is any answer. Okay, so I got two answers. One is saying it is number A, another is saying it is number B. Let me check A or B, which of the following is the correct answer for problem number 2. So the correct answer will be, it is number B. Transpeptidase is the enzyme that is involved in the inhibition, the mechanism of action of penicillin. So this is the structure of penicillin. You have to memorize this structure, okay, because it is very, very important. So this portion of the penicillin, as you can see, it has a bicyclic system where a four-member lactam ring is clubbed with a thiazolidine ring, okay, which is five-member. And this portion, this entire portion is known as 6-amino penicillinic acid or 6-APA, okay. And this have a acyl side chain here is present, and this R is if the R is CH2 phenyl, then it is known as the or benzyl penicillin, it is known as the penicillin G. And if it is OCH2 CH2OPH, then it is known as the phenoxymethyl penicillin or penicillin V. Okay. So what is the mechanism of action of penicillin? So the structure of cell wall, first we need to understand, okay. We have to understand what is the structure of the cell wall. So in order to understand penicillin's mechanism of action, we have first to look at the structure of the bacterial cell wall, the mechanism by which it is formed. Bacteria have cell walls in order to survive in a large range of environmental conditions such as varying pH, temperature and osmotic pressure. Without a cell wall, water would easily continually enter the cell as a result of osmotic pressure, causing the cell wall to swell and burst, which is known as lysis. This cell wall is very porous and does not block the entry of water, but it does not thus prevent the cell swelling. Animal cells do not have the cell wall, making it perfect target for the antibacterial agents such as the penicillin. Okay, so this thing is crucial. Okay, animal cells, they do not have a cell wall, making it perfect target for the antibacterial agents such as penicillins. Okay, the cell wall is a peptidoglycan structure. In other words, it is made up of peptide and sugar unit. The structure of the wall consists of the parallel series of sugar backbone containing two types of sugar. One is the N-acyl muramic acid or NAM. Okay, and another is the N-acyl glucosamine or NAG. These two things you have to remember. Okay, there are two type of sugar. One is NAM or N-acyl muramic acid, and another is NAG or N-acyl glucosamine. Peptide chains are bound to the NAM sugar, and interesting to note that the presence of the D amino acids in these chains. In human biochemistry, there is only L amino acid, whereas the bacteria have the racemase enzymes that convert L amino acid to D amino acid. Okay, so these points are very crucial. The bacteria have the racemase enzyme which convert the L amino acid to D amino acid. Okay. In the final stages of the cell wall biosynthesis, the peptide chains are linked together by the displacement of D-alanine from one chain by glycine to another. About 30 enzymes are involved in the overall biosynthesis of cell wall, but in the final cross-linking reaction, which is inhibited by the penicillin. Okay, the final cross-linking reaction of the cell wall synthesis is inhibited by penicillin. This leads to the cell wall framework for no longer interlinked. As a result, the wall became fragile and can no longer prevent the cell from swelling and bursting. The enzyme responsible for the cross-linking is known as the transpeptidase enzyme. Okay, so the enzyme responsible for the cross-linking is known as the transpeptidase enzyme. There are several types of enzymes which are vary in character from one bacterial species to another, but they are all inhibited by the various degrees of penicillin. So how it has happened? So as you can see, this is the active site of the transpeptidase enzyme. We have a serine and a lysine in the active site. The serine has a hydroxyl group which acts as a basically acts as the nucleophile and attacks on the carbonyl carbon of this beta lactam ring of penicillin, and as a result this ring opens and we have this kind of structure. Now because of this formation of this kind of structure, then now the active site is blocked, and since the active site is blocked, now the growing peptide chain, it cannot come here, it is blocked here, and because of that what happened, the final cross linking it is not happening here. That is why the cell wall, it remains fragile and easily the water can insert to the cell because of this fragile cell wall, and the lysis happen which destroys the bacterial cell. Okay, so this is all about how the penicillin is how the penicillin is exerting its antibacterial activity, what is the mechanism of action, and how the transpeptidase enzyme is responsible for all these things. Okay, so this was the problem number 2. So now is it clear to all, or is there any doubt? If you have any doubt, you can let me know. I will discuss it one more time. Otherwise, let me move to the next problem. So I have told you, if you have any doubt or query, you can let me know via chat box. Okay, anything, any question you do not understand, please let me know. Then and there, I will discuss it one more time. So if it is clear, then I will move to our next problem. Okay. So let us move to the next problem. So problem number 3, it says, which of the following is not an example of the anti-metabolites used? Which of the following is not an example of anti-metabolites used as the antibacterial therapeutics? Your options are number A, trimethoprim, number B, doxycycline, number C, sulfone, and number D, sulfomethoxazole. Which of the following is not an example of the anti-metabolites used as the antibacterial therapeutics? Your options are number A, trimethoprim, number B, doxycycline, number C, sulfone, and number D, sulfamethoxazole. Let me know A, B, C or D, which of the following will be correct answer for problem number 3. Again, I will wait for a minute or two, then I will check the chat box. Which of the following is not an example of anti-metabolized used as the antibacterial therapeutics? Number A, trimethoprim, number B, doxycycline, number C, sulfone, and number D, sulfamethoxazole. Which of the following is not an example of antimetabolites used as the antibacterial therapeutics? Trimethoprim, doxycycline, sulfone, or sulfamethoxazole? Let me check the chat box. Let me see. Okay, I got only one answer which is saying it is number A. Okay, let me check if number A is the correct answer for our problem number 3, or is there any different answer? So the correct answer for problem number 3 is, it is number B, doxycycline. Doxycycline is not an example of antimetabolites used as the antibacterial therapeutics. So here you can see, let me just use the highlighter. So here you can see the examples for the antimetabolites which are in medical use as the antibacterial agents. We have this trimethoprim, which is basically a antimalarial agent. We have sulfones as an anti-leprosy agent, and we have the sulfamethoxazole. Okay, so Trimethoprim is an orally active diaminopyrimidine structure which has proved to be highly selective antibacterial and antimalarial agent. It acts as the against the dihydrofolate reductase. We have actually discussed it in our for our class. I think it is our week 8 most probably. I have discussed that trimethoprim acts against the dihydrofolate reductase, an enzyme which carries out the conversion of dihydrofolate to tetrahydrofolate, leading to the inhibition of DNA synthesis and cell growth. Dihydrofolate reductase is present in the mammalian cells as well as in bacterial cells, but mutations over millions of years have resulted the significance difference in the structure between two enzymes such that trimethoprim recognizes and inhibits the bacterial enzymes more strongly. In fact, trimethoprim is 1 lakh times more active against the bacterial enzyme. Trimethoprim is often given in conjugation with the sulfonamide sulfamethoxazole in a preparation called clotrimazole. So this is important. Okay, trimethoprim is often given with the sulfonamide sulfamethoxazole, and the preparation is known as the clotrimazole. Clotrimazole. The sulfonamide inhibits the incorporation of para-aminobenzoic acid or PABA into the dihydrofolate, while trimethoprim inhibits the dihydrofolate reductase. Therefore, two enzymes in one biosynthetic route is inhibited. This is a very effective method of inhibiting biosynthetic route and has the advantage that the doses of both drugs can keep down the safe level. To get same level of inhibition using a single drug, the dose level would have to be much higher, leading to possible side effect. This approach is described as the sequential blocking. This is very important. The sequential blocking. So, what is the sequential blocking? The sequential blocking is to use two or more agents to stop a particular biosynthetic pathway. For example, this particular conversion of dihydrofolate to tetrahydrofolate, it can be inhibited by trimethoprim itself or by the sulfamethoxazole itself. Okay, by using one of them also can be done, but then the higher concentration of the drugs are required. But if we use together in a minimum level of concentration, then we will get a similar effect and it is safer. So this approach is known as a sequential blocking, where sequentially two different agents are incorporated to inhibit a particular biological pathway. Okay, so this is an example, very good example of sequential blocking. So the resistance to trimethoprim has been observed in certain strains of the E. coli which produce a new form of the target enzyme, and that has a less affinity to the drug. Like sulfones, the sulfones are most important drugs used in the treatment of leprosy. It is believed that they inhibit the same bacterial enzyme inhibited by the sulfonamides, like the, for example, which is the dihydrofolate reductase. So all these are the anti-metabolites that are used in the antibacterial therapeutics. Doxycycline is have a different kind of therapeutic effect. Okay, we will discuss about the effect of doxycycline in our upcoming classes. So this was the problem number 3. So now is it clear what is the role of the different antimetabolites in the antibacterial agent and how what is sequential blocking? So these things are very important. These are the small things which are not in very detail, but these are the very subtle things which you have to know, which you have to understand, and also which you have to remember. Okay. So now let me go to the next problem. If there is no issue with this issue problem, if you have any doubt, please feel free and let me know in the chat box. So problem number 4 says, which of the following statement is correct? Your options are number A, chloramphenicol is a broad spectrum antibiotic. Number B, it inhibits the bacterial DNA synthesis. Number C, it cannot be used topically. Number D, it contains two phenyl groups. Which of the following statement is correct regarding chloramphenicol? Your options are number A, chloramphenicol is a broad spectrum antibiotic. Number B, it inhibits the bacterial DNA synthesis. Number C, it cannot be used topically. And number D, it contains two phenyl groups. Which of the following statements is correct regarding chloramphenicol? Read all the options very carefully and then let me know A, B, C or D which of the following will be the correct statement regarding chloramphenicol. I will wait for 2 to 3 minutes again, or I will wait for a minute or two, then I will check the chat box. Okay. Meanwhile, let me know which of the following statement is correct regarding chloramphenicol. Your options are number A, chloramphenicol is a broad spectrum antibiotic. Number B, it inhibits the bacterial DNA synthesis. Number C, it cannot be used topically. Number D, it contains two phenyl groups. Which of the following statements is correct? A, B, C or D? Option number A, chloramphenicol is a broad spectrum antibiotic. Option number B, it inhibits bacterial DNA synthesis. Option number C, it cannot be used topically. Option number D, it contains two phenyl groups. What will be the correct statement? Let me check the chat box. Let me see if there is any answer for our problem number 4. Okay, I got an answer which says number C is the correct answer. Now let me check if number C is correct answer or not. So the statement which is correct will be, it is number A. Chloramphenicol is a broad spectrum antibiotic. This statement is correct. Okay, other statements are not. So this is the structure of chloramphenicol. So as you can see, the structure, it has a phenyl ring which has a nitro and a group of 1,4 position. Here we have a amide linkage is here present. Okay, so this is the structure of chloramphenicol. So the statement number D, which says it contains 2 phenyl ring, it is incorrect, right? So this statement is not correct. So okay, let me just not do it. Okay. So the statement number D is incorrect because it says it has 2 phenyl ring. No, it does not have 2 phenyl rings. So basically, chloramphenicol is a broad spectrum antibiotic used to treat serious bacterial infections such as meningitis, typhoid fever, and cholera. When safer antibiotics are not an option, its use is limited due to the high risks of severe and potentially fatal side effects, including bone marrow toxicity. So this is important. Chloramphenicol is used as a last minute option because it has a very high toxicity or side effects, including bone marrow toxicity. What is the mechanism of action? So chloramphenicol works by inhibiting bacterial protein synthesis. So the statement number B, which says it inhibits bacterial DNA synthesis, it is not correct. It inhibits bacterial protein synthesis. It binds reversibly to the 50S ribosomal subunit of bacteria, which prevents the peptidal transferase activity required for the creation of polypeptide chain. This interference stops the bacterial growth, and antibiotic is therefore primarily considered a bacteriostatic agent. However, chloramphenicol can have a bactericidal effect on certain organisms, such as thus causing the meningitis. Okay. Medical uses, because of the serious side effect, the use of the oral and intravenous. Okay, one second. Because of the serious side effect, the use of the oral and intravenous chloramphenicol is restricted to severe infection is limited to the with limited treatment alternatives. Systematic infection, it is used to treat serious life-threatening infection where resistance of major allergies prevent the use of other antibiotic. This includes certain types of meningitis, plague and rickettsial infections like Rocky Mountain spotted fever. Okay, and topical infection, chloramphenicol is still widely used as topical formulation like eye drops and ointment to treat the bacterial conjunctivitis and the superficial eye infection, where the risks of systematic effects are much lower. So the statement number C, which says it cannot be used topically, it is not correct because it can be used topically, and it is used topically as eye drops and ointments to treat the bacterial conjunctivitis. Okay, severe adverse effects, the risks of the severe adverse effect means the patient taking systematic chloramphenicol must be closely monitored. For the bone marrow suppression, this is a dose dependent effect on the bone marrow that cause the reversible decrease in the blood cell production. It is a common side effect of the systematic chloramphenicol use. Aplastic anemia, a rare but often fatal side effect of the chloramphenicol. It is not dose dependent and it can appear for weeks or months to treat has ended. The FDA has withdrawn all the oral chloramphenicol product in the United States due to the high risks of the aplastic anemia. Gray baby syndrome, a toxic reaction that causes newborn infants, particularly premature babies who have difficulty metabolizing and excreting the drug. This can cause abdominal bloating, a gray skin color, vomiting, low blood pressure, and cardiovascular collapse. Okay, so these are the severe adverse effects of the chloramphenicol. So, is it all right for the problem number 4, or is there any doubt? If not, then let me go to the next problem. If you have any query or any doubt regarding this problem, you can let me know in the chat box. Okay. Okay. Now let me go to the problem number 5 if there is no issue or no doubt. So problem number 5, it says, which of the following is an incorrect statement regarding the SAR of the penicillin? SAR means the structure activity relationship studies. Okay. So your options are: number A, beta lactam ring is essential. Number B, a bicyclic system is essential. Number C, methyl groups at C2 position are essential. Number D, free carboxylic acid group is essential. Which of the following is an incorrect statement regarding the SAR of penicillin? I have shown you what is the structure of penicillin, and I have said that it is very important to remember the structure of penicillin, because there are several aspects, and the SAR of penicillin is also very much important, which we are going to discuss now. But first, let me know A, B, C, or D. Which of the following you think is the correct answer of problem number 5? Which of the following you think is the incorrect statement regarding the SAR of penicillin? Your options are: number A, the beta-lactam ring is essential; number B, a bicyclic system is essential; number C, methyl groups in C2 positions are essential; and number D, free carboxylic acid group is essential. Which of the following is an incorrect statement regarding the SAR of penicillin? I will wait for a minute again, then I will check the chat box. Till then, let me know A, B, C, or D. Which of the following you think is the incorrect statement regarding the SAR of penicillin? Your options are: number A, beta-lactam ring is essential; number B, a bicyclic system is essential; number C, methyl groups at C2 position are essential; and number D, free carboxylic acid group is essential. Let me check the chat box. Okay, I got one answer which is saying that number D is the correct answer. Let me check if number D is the correct answer or not, and let us discuss about the SAR of penicillin. So this question is very important. Okay, the SAR of penicillin. So the correct answer means the statement which is incorrect will be, it is number C. Methyl groups in C2 position are essential. Now let me show you. So this is the structure of penicillin. Again, I am saying you have to memorize the structure of penicillin. I never say for memorizing a particular structure, but for penicillin you have to memorize the structure because it is very crucial. So as you can see, penicillin has a bicyclic ring. The system, okay, one is a lactam ring. So this lactam ring, and this is a 5-member thiazidinone ring. Okay. So this 5-member ring has a sulfur and nitrogen in 1 and 4 position. So numbering start from here 1, 2, 3. Then nitrogen is 4, 5, and there is 6. So this lactam ring is essential for the activity. Okay, this bicyclic ring system is also essential. This free carboxylic acid is essential. This cis stereochemistry is essential, and this amide is essential. So these are the essential things. Okay, and about the structure. The first is the lactam. This lactam ring is essential. Okay. Then this bicyclic system. The system must be bicyclic, otherwise it will not show any activity. The free carboxylate group is also essential. The COO minus group, it is essential. The cis stereochemistry is essential. This cis stereochemistry, it is also essential. And this amide linkage, it is essential. Okay. So the cis stereochemistry of these two hydrogens are essential. So this is the structure activity relationship relationships of the penicillin. So, the large number of penicillin analogues have been synthesized and studies. The results of these studies led to the following SAR conclusions. A strained beta-lactam ring is essential. The free carboxylic acid is essential. This is usually ionized. The penicillins are administered as sodium or potassium salts. The carboxylate ion binds to the charged nitrogen of the lysine residue in the binding site. the bicyclic. So if you remember that in the active site of the transpeptidase enzyme, we have a serine and we have a lysine. That lysine has a charged nitrogen, so that positive charge of the nitrogen is interacts with this negative charge of the carboxylate. Okay, the bicyclic, that is why this carboxylate is very crucial. The bicyclic system is important. It converts the further strain to beta lactam ring. The greater the strain, greater the activity, but greater the instability of the molecule, and other factors. Okay, sulfur is usual but not essential. The stereochemistry of the bicyclic ring with respect to the acyl amino side chain is important. The results of this analysis led to the inevitable conclusion that very little variation is tolerated by penicillin nucleus, but any variation are restricted to the acyl amino side chain. Okay, so the variations can be done in this acyl amino side chain, but not much variation can be done in case of your, okay, not much variation it can be done in our backbone on and or these kind of things. Okay, so is it clear? Again, I'm saying if you have any doubt, please let me know, or I will go to the next problem. Okay, whatever doubt you have, you can let me know. I can discuss the doubts at the end of the session also. Okay, so I think it is clear. So this SAR of penicillin is very important. Okay, this structure, you have to remember the structure, and also you have to remember the SAR means which part of the, so SAR structure activity relationship studies means which part of the, a particular part of the structure, a particular functional group, it how it contributes to the activity of that particular molecule. Okay, that is the structure activity relationship studies. Okay, so if it is clear, let me go to the next problem. So problem number 6 says pivamcillin is a prodrug of dash. You have to fill in the blanks. Your options are number A penicillin, number B cephalosporin, number C ampicillin, and number D amoxicillin. So pivamcillin is a prodrug of what? Penicillin, cephalosporin, ampicillin, or amoxicillin. In the last class, or the last to last class, I have discussed about the, I think in the last class, yeah, in the last class I have discussed about the prodrug therapy, why prodrugs are necessary, because sometimes it is not possible to administer the particular active form of the drug, and if we administer it, it may get inactive in the course of the administration or the course of the drug, till the drug is reaching to the active site, it may get inactivated. Okay, so that is why prodrugs are given, and the active site, the prodrugs are converted to the active form of drug, and it exerts this biological activity. So pivamcillin is a prodrug of what? Number A penicillin, number B cephalosporin, number C ampicillin, and number D amoxicillin. Again, I will wait for a minute or two, then I will check the chat box. Let me know A, B, C, or D, which of the following will be correct answer. Pivamcillin is a prodrug of what? Penicillin, cephalosporin, ampicillin, or amoxicillin. Now let me check the chat box. Let me check if there is any answer for our problem number 6. Okay, I got two answer, number D and number C for problem number 6. Now let me check number D or number C, which of the following will be the correct answer. Okay. Okay. 1 second. So the correct answer will be, so pivamcillin is a prodrug of, the correct answer is number C, ampicillin. Okay, it is a prodrug of ampicillin. So ampicillin prodrugs. So these are the structures of the, so this is the structure of the ampicillin. Okay, this is the structure of basic structure. Okay, and the R, this is the, these are the prodrugs. Okay, so this is R group is, if the R group is like this is known as the pivamcillin. Okay, if the R group is this, it is known as the talampicillin, and if the R group is this, so this is known as the vacampicillin. So these are this pivamcillin, talampicillin, and vacampicillin are the prodrugs of the ampicillin. These three cases, the esters used to mark the carboxylate group seem rather to elaborate, and one may ask why the simple methyl ester is not used. Okay, so why the simple methyl ester is not used? The answer is that methyl esters of the penicillins are not metabolized in human. The bulky penicillin skeleton is close to the ester, is so close to the ester that it acts as a steric shield and prevents the esterase enzyme that catalyze the reaction from accepting the penicillin ester as substrate. Fortunately, acyl methoxy methyl esters, this acyl methoxy methyl esters contain a second ester group for the further taking over the penicillin nucleus, which is more exposed to the attack. The hydrolysis products are inherently unstable and decompose spontaneously to release formaldehyde and reveal the free carboxylic acid. So how this, what is the mechanism of action, how this prodrug is activated to the drug? So this is, for example, this is the pivamcillin, right? So this is the pivamcillin. Now esterase, so all these groups, if you can see, all these have another ester group here present in its structure. So this is more exposed and esterase will attack here, and we will get this kind of structure which is unstable, and quickly it will release formaldehyde, then we will get the active drug. Okay. This release of the formaldehyde is not ideal. It is a toxic chemical. However, it is formed naturally in our body through enzymatic demethylation of various compounds found in diet, and levels is produced from the products described to cause little problem. Moreover, the drugs are only taken for the short period of time. Such extended esters can be used to prepare prodrugs of other penicillins, but one has to be more careful that one does not go to the other extreme and make the penicillin too lipophilic. For example, the one acyl methoxy ester of the penicillin G is so lipophilic that it has a poor solubility in water. Fortunately, the problem can be avoided easily by making the extended ester more polar. So this is the polar extended ester for the penicillin G. This valine, L-valine is added for increasing the polarity. Okay. So I think it is clear. So you have to remember the names of the prodrugs, and it is very good if you can remember the structures also, and also how the structures are activated to release the active drug from the prodrugs. Okay. So this was the problem number 6. Now let me go to the next problem, the problem number 7. So problem number 7 says augmentin, a life-saving drug, is a mixture of, your options are number A, oxacillin and ampicillin, number B, amoxicillin and clavuanic acid, number C, oxacillin and clavuanic acid, number D, amoxicillin and pyruvic acid. Augmentin, a life-saving drug, is a mixture of number A, oxacillin and ampicillin, number B, amoxicillin and clavuanic acid, number C, oxacillin and clavuanic acid, and number D, amoxicillin and pyruvic acid. Augmentin, a life-saving drug, is a mixture of what? Let me wait for a minute or two, then I will check the chat box. I think I have discussed about this mixture earlier also. Anyways, I will discuss it in detail in today's class. Augmentin, a life-saving drug, is a mixture of what? Number A, oxacillin and ampicillin. Number B, amoxicillin and clavuanic acid. Number C, oxacillin and clavuanic acid. Number D, amoxicillin and pyruvic acid. I will wait for a minute, then I will check the chat box. Let me know A, B, C or D, which of the following is the correct answer. Augmentin, a life-saving drug, is a mixture of what? Number A, oxacillin and ampicillin. Number B, amoxicillin and clavulanic acid. Number C, oxacillin and clavulanic acid. Number D, amoxicillin and pyruvic acid. Let me check the chat box. Let me see if there is any answer for our problem number 7. Okay, I got two answers. One is saying number A and another is saying number B. Let me check which of the following is the correct answer, number A or number B. Okay. So the correct answer, augmentin, is a mixture of, okay, let me use the highlighter. So it is number B, amoxicillin and clavulanic acid. So this is the structure of amoxicillin, okay, and this is the structure of clavulanic acid. So clavulanic acid was isolated. So amoxicillin, I have discussed it about amoxicillin in earlier slide. So clavulanic acid, now let us discuss about it. It was isolated from the S. clavuaris by Beckham's in 1976. It has a weak and unimportant antibiotic activity, but it has a powerful and irreversible inhibitor of most beta lactamases, which means it is used to as a sentry drug. So it is important. Okay, this clavulanic acid is used as a sentry drug in combination with the traditional penicillin. So why? Because this clavulanic acid is a powerful and irreversible inhibitor of most beta lactamases. This beta lactamases, actually what they do, they inhibits this beta penicillin by this enzymes. They actually break this lactam ring so that the antibiotic activity of penicillin or this beta lactam antibiotics, they are removed. Okay, so but this clavulanic acid is a powerful and irreversible inhibitor of the beta lactamases, which means can be used as sentry drug in combination with the traditional penicillin such as amoxicillin, and the combination of amoxicillin and this clavulanic acid is known as the augmentin, which is a life-saving mixture, and it is also sold in market as this augmentin mixture. Okay, this allows the dose levels of amoxicillin to be decreased and also increases the spectrum of activity. However, it should be noted there are various types of beta lactamases. Although the clavulanic acid is effective against the most of these, it is not effective against all. Clavulanic acid is also administered intravenously with tricursilin as the timentin. Okay, this is also important, that clavulanic acid not only with amoxicillin, it is also administered but intravenously with tricursilin, and the mixture is known as a timentin. So the structure of clavulanic acid was first example of the naturally occurring compound where beta lactam ring is not fused with a sulfur containing ring. Instead, it is fused with a oxalidione ring. So as you can see here, the beta lactam, there is no sulfur. Here sulfur is replaced by oxygen. The structure is also unusual in that it does not have any acyl amino side chain. So you see there is no acyl amino side chain. Many analogues have now been made. An essential requirement of the beta lactamase inhibition is the strain beta lactam ring. Okay, so beta lactam ring must be present. The enol ether, okay, the enol ether, this Z enol ether, the Z configuration of the double bond of enol ether is also required. Activity is reduced but not eliminated if the double bond is E. No substitution at is 6, so at a position 6 there is no acyl amino side chain. Okay, no substitution at the carbon 6. R stereochemistry of position 2 and 5. Okay, so position 2, this position and 2 and 5. So there you can see this hydrogen and this carboxylate both have the R stereochemistry. The carboxylate group is necessary. It is also thought that the 9-hydroxyl group, this 9-hydroxyl group is involved in the hydrogen bonding interaction with the active site of beta lactamase. Clavulanic acid is a mechanism based inhibitor and is classified as a suicide substrate. So I think when I was discussing about the suicide substrates and the suicide enzyme inhibition in our second to third class, or maybe it can be fourth class also. So I think that time I have discussed about the clavulanic acid, because I am remembering that I have discussed about this clavulanic acid and this augmentin. Okay, so this is the SAR of clavulanic acid, and this is so what is the role of the, so it is a very good example of a sentry drug where it is used in addition with an traditional penicillin like amoxicillin or tricursilin. Okay, so is it clear the SAR of the clavulanic acid, or do I need to repeat it? I hope it is clear. Okay, if it is not clear, please let me know. I will discuss it one more time. These are very simple. Once you have to read the things and then you have to practice. Okay, this is I think that that will be fine if you do these two things together. Anyways, if you have any problem, you can let me know via email also, or you can let me know in the next class, or you can let me know via the discussion forum. Now, let us move to the next problem. Problem number 8 says that IPN to 6 APA conversion is done by your option are number A, ACVS, number B, IPNS, number C, DAOCS, and number D, IPNA. IPN to 6 APA conversion is done by which enzyme? ACVS, IPNS, DAOCS, or IPNA? Let me know A, B, C or D which of the following will be the correct answer of problem number 9. Again, I will problem number 8. Again, I will wait for a minute or two. Then I will check the chat box. Meanwhile, let me know A, B, C or D which of the following will be the correct answer of problem number 8. IPN to 6-APA conversion is done by which enzyme? Number A, ACVS, number B, IPNS, number C, DAOCS, and number D, IPNA. IPN to 6-APA conversion is done by which enzyme? Let me check the chat box. Let me check the answers. Okay, I got one answer which is saying number B is the correct answer or number B is the correct enzyme. Okay, let me check if number B is the correct answer or not. So IPN to 6-APA conversion is done by, it is number D, IPNA. Okay, so now let us discuss what are these abbreviations mean. Okay, so first is our ACVS means the ACV synthase, or ACV means the delta L alpha amino adipoyl L cysteine D valine. Okay, so first this is the biosynthesis of the penicillin. So it starts with the L alpha amino adipic acid, L lysine and L valine. So first it is converted to the alpha amino adipoyl L cysteine D valine or ACV by ACV synthase. Okay, and then ACV synthase is converted to the IPN or isopenicillin N by IPNS. Okay. Then IPN is converted to 6-APA by IPN amino hydrolase or acyl transferase or IPNA. Okay, then it is converted to other penicillin. And this IPN epimerase converts IPN isopenicillin N to penicillin N, and which is also converted to the deoxyacetylate cephalosporin C by DOCS or DAOC synthase. So IPNA catalyzes the complex two-step cyclization reaction. So as you can see, okay, so it catalyzes the two-step cyclization process, and this converts the linear tripeptide substrate, this linear into the bicyclic product of the IPN. Okay, a second. Yeah, oxygenase activity. The enzyme is a non-heme protein belonging to two auxiliary dependent deoxys family. It uses a unusual oxygenase activity to form both beta-lactam and thiazolidine ring found in the penicillin structure. Active site, the active site of the IPNS. Okay, this is about the IPNS. Okay, so IPNS contains the ferrous. So I think there is a little, I mean, this thing is not correct. Okay, let me just delete it. I think I have mixed the two things together. Okay, So IPN to 6-APA conversion is done by IPN-A. That slide I have mistakenly added. So what is the biological significance of IPN-A? So IPN-A is an early and crucial enzyme in the pathway for the producing all the penicillin and cephalosporin. There are among the most widely used antibiotics in the world. Gene regulation, the gene that codes for IPN-A known as the IPN-A. This is the here IPN is in lowercase and A is the uppercase and all are the italics. It is regulated by the ambient pH condition in the fungi like the Aspergillus nidulans. Okay. So remember, IPN to 6-APA conversion is done by IPNA or IPN amino hydrolase or acyltransferase. Now let me go to the next problem. Problem number 9 says D-cycloserine exerts its antibacterial action by inhibiting. Your options are number A, L-alanine racemase, number B, carboxypeptidase, number C, carbonic anhydrase, and number D, L-serine racemase. D-cycloserine exerts its antibacterial action by inhibiting which of the following enzymes? Your options are number A, L-alanine racemase, number B, carboxypeptidase, number C, carbonic anhydrase, and number D, L-serine racemase. D-cycloserine exerts its antibacterial action by inhibiting L-alanine racemase, carboxypeptidase, carbonic anhydrase, or L-serine racemase. Let me know A, B, C, or D which of the following will be the correct answer for problem number 9. Let me wait for a minute or two again and then I will check the chat box. Meanwhile, let me know. D-cycloserine exerts its antibacterial action by inhibiting which enzyme? L-alanine racemase, carboxypeptidase, carbonic anhydrase, or L-serine racemase? Let me check the chat box. Let me see if there is any answer for problem number 9. Okay, I got two answers. One is saying number D, another is saying number C. Let me know. Let me check number D or number C. Which of the is following is the correct answer, or is there any other answer which is correct? So for number 9, the correct answer is it is number A, L-alanine racemase. It is the enzyme that is inhibited by D-cycloserine when it is showing its antibacterial action. So the mechanism of action, cycloserine is a structural analog of amino acid D-alanine and functions as a competitive inhibitor of two key enzymes of the of our cell wall synthesis. Okay, so what are the two key enzymes? The target enzymes is the D-alanine racemase. Okay, okay, sorry, it will be deep alanine racemase, not L-alanine. A second. Okay, so let me just correct it. Okay, so sorry, it was a little mistake here. So it is the target enzyme for our this D-cycloserine is the D-alanine racemase and the D-alanine ligase. Okay, D-alanine ligase. So this inhibition pathway, these two enzymes are crucial for creating the peptidoglycan component in the bacterial cell wall. D-alanine racemase converts L-alanine to D-alanine and D-alanine ligase joins two D-alanine residues. Outcome: by blocking this step, cycloserine prevents the formation. So the cycloserine prevents the formation of the functional cell wall, causing the bacteria to weaken and die. Primary uses: the tuberculosis, cyclosporin is used in combination with other anti-TB medications, especially for the multi-drug resistant TB or MDR TB, where the first line treatments are ineffective. Urinary tract infection, in some cases, it can be used to treat UTIs caused by the susceptible bacteria when the other treatments have failed. Side effects, cyclosporin treatment is associated with significantly neuropsychiatric side effects which can limit its use. The effects are often dose dependent. Common side effects: anxiety, confusion, drowsiness, dizziness, mood or mental changes. A severe side effect: convulsions or seizures, depression, psychosis, aggression, suicidal thoughts can occur. Other side effects: headache, numbness, tingling in the hands, feet, peripheral neuropathy, and the speech problems. Contradictions: because of these effects, cyclosporin is not recommended for the people with a history of the seizure disorder, severe kidney disease, or mental illness. Brand names: cycloserine is available in the generic form, and some brand names include the seromycin and coxerine. The dosage is usually administered as an oral capsule. Dosage is determined by the doctor and depends on the patient's condition and severity of the infection. The effectively treat the TB, it's crucial for patient complete to full course of treatment even if they start feeling better. So remember, the target enzymes for the cycloserine is the D-alanine racemase and D-alanine ligase. The D-alanine racemase converts L-alanine to D-alanine, and D-alanine ligase join two D-alanine residues. So by blocking this, cycloserine prevents the formation of the functional cell wall, causing the bacteria to weaken and die. Okay. So is it clear? So there was a little, mistake in the typographical error in the answer, which I have corrected now. Okay. So now I will go to the next problem. So problem number 10, it says that, okay, just wait. The transformation of penicillin to sulfoxide to cephalosporin, the penicillin sulfoxide to cephalosporin is termed as number A, Pomeroy rearrangement, number B, Morin rearrangement, number C, Schmidt rearrangement, and number D, Claisen rearrangement. The transformation of penicillin sulfoxides to cephalosporins is termed as Pomeroy rearrangement, Morin rearrangement, Schmidt rearrangement, or Claisen rearrangement. Let me know A, B, C, or D, which of the following will be the correct answer for problem number 10. The transformation of penicillin sulfoxide to cephalosporin is termed as which rearrangement? Pomeroy rearrangement, Morin rearrangement, Schmidt, or Claisen? Again, I will wait for a minute or two, then I will check the chat box. Okay. The transformation of penicillin sulfoxides to cephalosporin is termed as which rearrangement? The transformation of penicillin sulfoxides to cephalosporin is termed as which rearrangement? Pomeroy rearrangement, Morin rearrangement, Schmidt, or Claisen rearrangement? I will wait for a minute and then I will check the chat box. So the transformation of penicillin sulfoxide to cephalosporin is termed as what? Which rearrangement will be the correct answer? Let me check the chat box. Okay, so I got one answer which is saying that number A is the correct answer. Now let me check. Sorry. Now let me check if number A is the correct answer or not. Okay. Okay. So the correct answer will be, it is number B, Morin rearrangement. Okay. So sulfonic acids or RSOH, these are among the most common sulfur containing reactive intermediate generated in the biological systems. Okay, so the sulfonic acids or RSOH are the most common sulfur centered reactive intermediate generated in biological system. Given the biological occurrence of the sulfonic acids, it is important to explore the reactivity of these intermediates under the physiological conditions. The Morin rearrangement a synthetic process to develop the conversion of penicillin derivative into the cephalosporin that proceed via a nucleophilic attack on the alkene on the sulfonic acid intermediate. Okay, so this is important. Moric acid rearrangement is a synthetic process developed for the conversion of penicillin derivatives into cephalosporins that proceeds via a nucleophilic attack of an alkene on a sulfonic acid intermediate. In the classic form, the Morin reaction involves initial elimination of the sulfonic acid from a cyclic sulfoxide followed by intermolecular cyclization of the resulting alkene and sulfonic acid groups to generate an episulfonium ion intermediate that undergoes further reaction to yield the ring expanded products. On the basis of the existing literature, it is difficult to assess whether the reaction between the alkene and the sulfonic group can occur under the mild condition because the condition required to generate the sulfonic acid from the sulfoxide precursor in the Morin reaction typically involve high temperatures and strong acid. In the work described here, so this is the thing, okay. So remember, so you do not need to memorize much from this. Just you have to remember that Morin rearrangement is a reaction which is involved in the conversion of the penicillin derivatives to the cephalosporins. Okay. Now I will go to the next problem. Problem number 11 says, which of the following antibacterial agents work by affecting the bacterial plasma membrane permeability? Your options are number A, polymyxin, number B, penicillin, number C, aminoglycoside, and number D, nalidixic acid. Which of the following antibacterial agents work by affecting the bacterial plasma membrane permeability? Your options are number A, polymyxin, number B, penicillin, number C, aminoglycoside, and number D, nalidixic acid. Let me know A, B, C, or D. Which of the following is the correct answer for problem number 11? Which of the following antibacterial agents work by affecting bacterial plasma membrane permeability? Polymyxin, penicillin, aminoglycoside, or nalidixic acids? I will wait for a minute and then I will check the chat box. Okay. Till then, let me know A, B, C, or D. Which of the following you think is the correct answer for problem number 11? Which of the following antibacterial agents work by affecting the bacterial plasma membrane permeability? Let me check the chat box. Let me see if there is any answer for problem number 11. Okay, I got an answer which saying that number C is the correct answer. Let me check if number C is the correct answer or not. So the agent which exerts this antibacterial activity by affecting bacterial plasma membrane permeability will be number A, polymyxin. Okay, so this figure is a very good to remember and to understand. So so this is the bacterial cell. Okay, and what are the drug targets? Now is this is the cell. This is the outer membrane. Okay, this is the outer membrane. This is the periplasmic space. This is the cell wall. Okay, and this is the cytoplasm. So in the cytoplasm we have this nuclear material like DNA or RNA. We have ribosomes. Okay, we have this, and this is the plasma membrane. So as you can see, first we have the outer membrane followed by a periplasmic space, then cell wall, then we have a plasma membrane. Plasma membrane is is outside the cytoplasm. So now the if what are the agents that affect the thing? So we have the penicillin, cephalosporin, and cyclosporin. They affect the cell wall synthesis. We have sulfonamide. They target the which targets the enzymes. We have rifamycin, which targets the nuclear material. So as the quinoline and amyloacridins, they also target the nuclear material. Chloramphenicol, streptomycin, tetracycline. These affect the ribosomes and protein synthesis. And polymyxin, as you can see, they affect the plasma membrane permeability. So the mechanism of action, mechanism of bacterial action. So these are the five main mechanisms. So there are five main mechanisms by which the antibacterial agents act. One is the inhibition of cell wall. So antibacterial agents which inhibit the cell wall metabolism are known as the antimetabolites. Okay, these compounds inhibit the metabolism of the microorganism but not the metabolism of the host. They can do it by inhibiting an enzyme catalyzed reaction which is present in bacterial cell but not in the animal cell. The best known example of antibacterial agent acting this way are sulfonamides. Okay, so the example of antimetabolites is the sulfonamides. Now, it is also possible for antibacterial agents to show the selectivity against the enzyme which is present in both bacterial and mammalian cell as long as there are two significant difference in the structure between these two. Then we have the inhibition of bacterial cell wall synthesis which leads to bacterial cell lysis and death. Agents so operating in this way include the penicillin, cephalosporin, glycopeptides such as vancomycin. As animal cells do not have any cell wall, they are not affected by these agents. Okay, so this is important that animal cells or human cells, they do not have a cell wall. That is why they are unaffected by this agent which targets the bacterial cell wall synthesis. Interactions with the plasma membrane. Okay, interactions with the plasma membrane. Some antibacterial agents interact with the plasma membrane of the bacterial cells to affect the membrane permeability. This has a fatal result. Polymyxins and tyrosine operate in this way. Disruption of the protein synthesis means the essential proteins and enzymes required for cell survival can no longer be made. Agents that which disrupt the protein synthesis include the rifamycin, aminoglycoside, tetracycline, and chloramphenicol. So they target the bacterial protein synthesis and inhibition of the nucleic acid transcription, replication, and cell division. Okay, so this is another way. So these are the agents that work in this way are the nalidixic acid and proflavin. So the five mechanism by which the antibacterial agent work is the inhibition of cell metabolism, inhibition of cell wall synthesis, disruption of protein synthesis, disruption of nucleic acid transcription replication, and interaction with the plasma membrane. And there are examples. So what you have to do is you have to remember all the mechanisms. Okay, what are the mechanism, how it works, and you have to remember the examples of the antibacterial agents by which it shows this particular effects. Okay, these things which you have to remember. Okay, this is very important. What are the different mechanisms and the examples of different antibacterial agents that work by this mechanism, the examples, these are the very crucial topics which you have to remember and which you have to understand. And this is the structure of the polymyxin. This is a very complicated structure which you do not remember, which you do not need to memorize. Okay, I have just given for your understanding. Now let me go to the next problem. So problem number 12, it says that telithromycin belongs to dash class of antibiotic. Your options are number A macrolide, number B tetracycline, number C oxylidinone, number D quinolone. Telithromycin belongs to which class of antibiotic? Number A macrolide, number B tetracycline. So it will be tetracycline. There is a spelling mistake. Okay. Number C oxylidinone and number D quinolone. Telithromycin belongs to which class of antibiotics? Macrolide, tetracycline, oxalidione or quinolone? Think and let me know in the chat box A, B, C or D which of the following will be the correct answer for problem number 12. Telithromycin belongs to which class of antibiotics? Macrolide, tetracycline, oxalidione or quinolone? Telithromycin belongs to which class of antibiotics? Let me check the chat box. Let me see if there is any answer for the problem number 12. Okay. I got one answer which is saying that it is number B. Let me check if number B is the correct answer or not. Okay. So telithromycin belongs to the correct answer is number A, macrolide. Okay. So this there is a spelling mistake. Let me correct it first. So it will be tetracycline. Yeah. So the correct answer is number A, which is macrolides. Okay. Let me discuss it. So, telithromycin, it belongs to the macrolide class of antibiotics. Okay. So macrolides are the okay. Let me just show you the structures also. So this is the this is the macrolide class of the bacteriostatic agents. Okay, this is the structure of the erythromycin, and this is the structure of the telithromycin. This is the structure of the azithromycin. Macrolides are the bacteriostatic agents. The best known example of this class of compounds is erythromycin, a metabolite isolated in 1952 from the soil microorganism Streptomyces erythraeus found in Philippines and one of the safest antibiotics in clinical use. The structure consists of the 14-member macrocyclic lactone ring with a sugar and an amino sugar attached. The sugar residues are important for the activity. Now, erythromycin acts by binding to the 50S subunits of the bacterial ribosome and inhibit the translocation, but other mechanisms are also appear to be likely because erythromycin and chloramphenicol bind to the same region or of the ribosome. They should not be administered together or they will compete with each other and be less effective. Erythromycin is unstable to stomach acids and can be taken orally in a tablet form. The formulation of the tablet involves the coating that is designed to protect the tablet during the passage through the stomach, but which is also soluble once it reaches the intestines. The enterosoluble. The acid sensitivity of erythromycin is due to the presence of the ketone and two alcohol groups, which are set for the acid catalyzed intermolecular formulation of the ketal. Now, one way to preventing this is to protect the hydroxyl groups. For example, calithromycin is a methoxy analog of erythromycin, which is more stable in gastric juices and improved oral absorption. Another method is increasing the acid stability to increase the ring size to the macrocycle of 16-member ring. Azithromycin contains 15-member macrocyclic, where the N-methyl group has been incorporated into the microcycle. One of the world's best-selling drugs is azithromycin. Telithromycin is a semi-synthetic derivative of erythromycin and reached the European market in 2001. The clandinose sugar in erythromycin has been replaced by the keto group and carbamate ring, then fused with the macrocyclic ring. Two hydroxyl group that cause intermolecular ketal formation in erythromycin has been masked, one with methoxy and another part of the carbamate ring. So this is the azithromycin. This is the, I think, one of all we have taken the azithromycin whenever we feel sick or we have high fever or bacterial infection, different kinds of. So we use this azithromycin. Then we have this telithromycin. This is the telithromycin, and this is the structure of erythromycin and calithromycin. All are these are examples of the macrolide class of compound, and these are the bacteriostatic agents. Okay, and this is the structures are very little complicated, so you do not need to memorize the structure, just you have to remember what is the mechanism of action of this kind of antibiotic agents. Okay, so now let me go to the next problem. Problem number 13 says how oxalidione works. Option number A, it inhibits the protein synthesis by binding to 30S ribosome. Number B, it inhibits the protein synthesis by binding to 50S ribosome. Number C, it unwinds the DNA double helix. And number D, it inhibits the DNA guidage enzyme. How oxalidione works? Your options are: number A, it inhibits the protein synthesis by binding with the 30S ribosome. Number B, it inhibits the protein synthesis by binding to 50S ribosome. Number C, it unwinds the DNA double helix. And number D, it inhibits the DNA guidase enzyme. Which of the following is the correct answer for problem number 13? Again, I will wait for a minute or two, then I will check the chat box. Meanwhile, let me know A, B, C or D, which of the following is the correct answer. How oxalidione works? Your options are: number A, it inhibits the protein synthesis by binding to 30S ribosome. Number B, it inhibits the protein synthesis by binding to 50S ribosome. Number C, it unwinds the DNA double helix. Number D, it inhibits the DNA guidage enzyme. Let me check the chat box. Okay, I got one answer which is saying number B is the correct answer. Let me check if number B is correct answer or not. So the correct answer will be yes, it is number B. It inhibits the protein synthesis by binding to the 50S ribosome. So oxalidione, these are the structures of the oxalidione. So these have a broad spectrum of activity and are against the bacterial strain and are active against bacterial strains which have acquired resistance to the other antibacterial agents. So, okay, linazolide has a good activity against the most clinically important gram-positive bacteria, including MRSA, and can be taken orally with 100% uptake from the gastrointestinal tract. And unfortunately, there is a high level of side effects related to the use, and it is bacteriostatic agent. There is a higher risk for bacterial resistant development. So this is the linazolide, and this is the redazolide. So these are the oxalidione. So this particular ring, which is the 5-member ring having a nitrogen and oxygen in 1 and 3 position along with a ketone group, this ring is known as the oxalidione ring. Okay. So this is a oxalidione ring, and since this particular antibacterial agents have this ring, these are known as the oxalidinones. So oxalidinones are the class of synthetic antibacterial agent discovered in recent years. They inhibit the protein synthesis by in much earlier stage than the previous agent, and consequently do not suffer from the resistance problem before protein synthesis can start. A 70S ribosome has formed in combination of 30S and 50S ribosome. The oxalidinones, they bind to the 50S ribosome and prevent this from happening. Okay, so so first we know that in the prokaryotic ribosome we have two subunits, one is 30S and one is 50S, and in the eukaryotic we have a 80S ribosome consisting of a 60 and 40S ribosomal subunits. So in oxalidinone, what they do, they bind to the 50S ribosome subunit and they prevent this 70S ribosome formation. As a result, translocation cannot start. Okay, translation cannot start. Other agents that inhibit protein synthesis by DOE is the translation process itself. Linazolide was one of the first class of this compound to reach the market in 2000, and by 2010 it was netting the sales like 16 million per year. here. And X-ray crystallographic studies have revealed that how the structure binds to the ribosome, and it has allowed the development of analogs bind more strongly. Radizolate is the one of the structure which binds 10,000 times more strongly as a result of extra binding interaction, and it is currently undergoing the clinical trials. Okay, so this is the all about the oxalidinones. Again, I am saying if you have any doubt or any query, please let me know through the chat box. Okay. So this is was the mechanism of action, how oxalidinone class of antibacterial agents they work. Okay. So now if there is no doubt, I will go to the next problem. So problem number 14, it says that which of the following is a general mechanism of action for vancomycin? Your options are number A, inhibition of metabolic enzyme, number B, inhibition of cell wall synthesis, number C, disruption of protein synthesis, and number D, inhibition of the nucleic acid transcription and replication. Which of the following is the general mechanism of action for vancomycin? Your options are number A, inhibition of metabolic enzyme, number B, inhibition of cell wall synthesis, number C, disruption of protein synthesis, number D, inhibition of nucleic acid transcription and replication. Which of the following is the general mechanism of action of for the vancomycin? Which of the following is the general mechanism of action for vancomycin? Your options are number A, inhibition of metabolic enzymes, number B, inhibition of cell wall synthesis, number C, disruption of protein synthesis, and number D, inhibition of nucleic acid transcription and replication. Let me wait for a minute, then I will check the chat box, and then we will discuss about the mechanism of action of vancomycin. I think I have discussed a little bit about the vancomycin in our last class also when we were discussing those things. I think I have discussed about it a little bit. Okay, today I will discuss it in detail. Okay, let me check the chat box. Let me check if there is any answer for problem number 14. Okay, I got one answer saying number D is the correct answer for problem number 14. Let me check if it is so. So, the correct answer will be, it is number B, inhibition of the cell wall synthesis. So this is the structure of the vancomycin. Again, it is a very complicated structure which you do not need to remember. Okay. So vancomycin, what is the mechanism of action? They they actually disrupt the membrane integrity by inhibiting the cell wall synthesis. Okay. Vancomycin is a crucial drug of the last resort, inhibits the peptidoglycan synthesis by the binding directly to the D-alpha DLI end of the peptide. Okay, so the D-alpha DLI end of the peptide, it is bound to by the vancomycin, and that's how it inhibits the peptidoglycan synthesis. It forms a cap over the end of the chain, blocking the cross-linking. So the final cross-linking, when all these peptides are formed by the transpeptidase and transglycolation, the resistant of this peptidoglycan, this cell wall is synthesized by vancomycin. They will create a cap, and they're susceptible to osmotic pressure and cell lysis, inhibits the peptidoglycan biosynthesis of the bacterial cell wall, blocks the transglycolase and transpeptidase activity, prevents the transpeptidation linking, stops the bacterial cell wall mutation. Okay, so this is the mechanism of action of vancomycin, how it exerts its antibacterial effect. Now, I will go to the next problem. Problem number 14 says dash acts as a starting material of 3-methylated cephalosporin. Your options are number A, sulfonamide, number B, streptomycin, number C, penicillin, number D, erythromycin. Which of the following acts as a starting material of the 3-methylated cephalosporin? Number A, sulfonamide, number B, streptomycin, number C, penicillin, and number D, erythromycin. Let me know A, B, C or D, which of the following is the correct answer for problem number 15. Dash acts as a starting material of 3-methylated cephalosporin. Which of the following will be the correct answer? Sulfonamide, streptomycin, penicillin or erythromycin? I will wait for a minute, then I will check the chat box, okay? Dash acts as a starting material of 3-methylated cephalosporin. What will be the correct answer? Sulfonamide, streptomycin, penicillin or erythromycin? Let me check the chat box. Okay. I don't have any answer for this problem. Okay, I got one answer, number A. Okay, let me check if number A is the correct answer or not. So the correct answer of problem number 15 will be, it is number C. Penicillin acts as a starting point or the starting material for the 3-methylated cephalosporin. So cephalexin is a methylated substituent at the position 3, okay, this which appears to help the oral absorption. A methyl group would normally be bad for the activity as it is not a good leaving group. However, the presence of the hydrophilic amino group in the alpha carbon of the 7-acyl amino side chain of cephalexin helps to restore the activity, and cephalexin is one of the few cephalosporin which is orally active. The mechanism of absorption through the gut wall is poorly understood, and it is not clear why 3-methyl group is advantageous for the absorption. Okay, so synthesis of 3-methylated cephalosporin involves the use of the penicillin as a starting material. So how it happens? This is shown in this scheme. So this is the penicillin. Okay, and first is the use of the hydrogen peroxide, so which will create a S plus O minus here. Then toluene and PTSA will lead to this ring opening for this thiazolidinone ring, and then followed by a ring expansion, which makes this 5-member ring a 6-member ring. Okay, and then other mechanism follows. Okay, so this is the mechanism how the synthesis, which is first demonstrated by E. Lilly pharmaceutical, involves the oxidation of sulfur by the acid catalyzed ring expansion, where 5-member ring thiazolidinone ring thiazolidine ring of penicillin is converted to 6-member dihydrothiazolidine ring in cephalosporin. Okay, the 5-member ring is known as the thiazolidine ring in penicillin, and the 6-member ring in cephalosporin is known as the dihydrothiazine ring. Okay, and cephalexin, this is the cephalexin is this is the structure of cephalexin. It has a 3-methyl substituent which is which appears to be very important for the biological activity, but why it is so, it is not clearly known till now. Okay, and this is the synthetic pathway where the 3-methylated cephalosporin are synthesized from penicillin. Okay. Now I will go to the next problem. Problem number 16 says rifampicin binds to dash. Fill in the blanks. Your options are number A, RNA independent DNA polymerase, number B, RNA dependent DNA polymerase, number C, DNA independent RNA polymerase, and number D, DNA dependent RNA polymerase. Rifampicin binds to which of the following enzymes? The options are number A, RNA dependent RNA independent DNA polymerase, number B, RNA dependent DNA polymerase, number C, DNA independent RNA polymerase, and number D, DNA dependent RNA polymerase. Rifampicin binds to which of the following enzymes? Again, I will wait for a minute, then I will check the chat box. Let me know A, B, C or D which of the following is the correct answer for problem number 16. Rifampicin binds to which enzyme? RNA independent DNA polymerase, RNA dependent DNA polymerase, DNA independent RNA polymerase, or DNA dependent RNA polymerase? Rifampicin binds to which enzyme? Let me check the chat box. Okay, I got an answer which says number B is the correct answer. Let me check if number B is correct answer or not. So rifampicin bind to it is number D, DNA dependent RNA polymerase. Okay, so rifampicin. So this is the structure of rifampicin. Okay, let me just use the highlighter. So this is the structure of rifampicin. Okay, so there is a functional group R is here present here. So if R is hydrogen, it is known as the rifampicin B. If R is this group, then it is known as the rifampicin, only rifampicin. Okay, rifamycin. So rifamycin, and this is the rifamycin. The structure is the rifamycin. So rifampicin is a semi-synthetic synthetic rifamycin made from the rifamycin B, an antibiotic which is isolated from the Streptomyces meditere in 1957. It inhibits the gram-positive bacteria and works by binding to the no covalently with the DNA-dependent RNA polymerase. Okay, it binds non-covalently with the DNA-dependent RNA polymerase and inhibiting the start of the RNA synthesis. The DNA-dependent RNA polymerase in eukaryotic cells are unaffected because the drug binds to the peptide chain is not present in the mammalian RNA polymerase. It is therefore highly selective. The flat naphthalene ring, the several of the hydroxyl groups and are essential for its activity. Okay, this naphthalene ring and several hydroxyl groups. So as you can see, there are several hydroxyl groups. These are the so this is also a kind of SAR. Okay, that the flat naphthalene ring and several hydroxyl groups are essential for the activity, and the molecule exists in the Zwitter ion, giving it a good solubility in both lipids and the aqueous acid. Okay. Rifamycin is another semi-synthetic analog that was approved in 2004 for the treatment of diarrhea and E. coli infection. So, rifamycin binds with this enzyme DNA-dependent RNA polymerase. Okay. Now let me go to the next problem. Problem number 17 says which of the following statements regarding Pseudomonas aeruginosa is false. Your options are number A, this is an example of opportunist pathogen. Number B, the cells of the P. aeruginosa are cone shaped. Number C, it prefers to grow in moist environment. Number D, it has intrinsic resistance to the wide variety of antibiotics. Which of the following statements regarding Pseudomonas aeruginosa is false? Your options are number A, this is an example of the opportunistic pathogen. Number B, the cells of the P. aeruginosa are cone shaped. Number C, it prefers to grow in moist environment. Number D, it has an intrinsic resistance to a wide variety of antibiotics. Which of the following statements regarding Pseudomonas aeruginosa is false? So again, it is a true false kind of question. So read the options very carefully. Again, I will wait for a minute or two and then I will check the chat box. Okay. Read the options carefully and then let me know A, B, C or D which of the following you think is the correct answer for problem number 17. Which of the following statements regarding Pseudomonas aeruginosa is false? Number A, this is an example of opportunistic pathogen. Number B, the cells of P. aeruginosa are cone shaped. Number C, it prefers to grow in the moist environment. And number D, it has intrinsic resistance to a wide variety of antibiotics. I will wait for a minute, then I will check the chat box, read the options carefully and let me know A, B, C or D which of the following is the correct answer. Which of the following statements regarding Pseudomonas aeruginosa is false? Let me check the chat box. Let me check if there is any answer for problem number 17. Okay, I got one answer which is saying that it is number B. Okay, let me check if number B is the correct answer or not. So the statement which is false regarding the Pseudomonas aeruginosa will be, it is number B. The cells of aeruginosa are cone-shaped. So this Pseudomonas aeruginosa is responsible for various diseases like catheter-related infection, bladder catheter infection, urinary tract infection, skin-borne infections, implant-associated infections, osteomyelitis, and pneumonia. Okay. So, the Pseudomonas aeruginosa is an example of the opportunistic pathogen. Such organisms are not normally harmful to healthy individuals. Indeed, many people carry the organism without being aware of it because their immune system keeps it under control. Once the immune system is weakened, though the organism can start the weakened, though the organism can start multiplying and lead to the serious illness. Hospital-bound patients are particularly at risk, especially those who are suffering from a shock or AIDS or those undergoing a cancer chemotherapy. Burn victims are particularly prone to pre-aeruginosa skin infections, and this can lead to septicemia, which can be fatal. The organism is also responsible for serious lung infection among the patients undergoing the mechanical ventilation. The cells of the P. aeruginosa are rod-shaped and can appear blue or green in color, which is why it is given the name of aeruginosa. This prefers to grow in the moist environment and can be isolated from soil, water, plants, and animals or humans. It can also even grow in the desired water, distilled water and contact lenses solutions. In hospital, there are several possible sources for the infection, including the respiratory environment, sinks and uncooked vegetables or flowers that brought by visitors. Pseudomonas aeruginosa is difficult organism to treat because it has an intrinsic resistance to wide variety of antibacterial agent, including many penicillin, cephalosporin, tetracycline, quinolones, chloramphenicol. There are two reasons for this. The outer membrane of the cell has a low permeability to the drugs. Even if the drug enters the cell, there is a efflux system which can pump it back outside. Nevertheless, there are drugs which have proved to be effective against the organism, in particular aminoglycosides such as tobramycin, gentamicin, penicillin such as ticarcillin. These are the combination can be act together. Okay, and this is the structure of the tobramycin. Now let me go to the next problem. Problem number 18 said aminoglycosides work by blocking dash during the translation. Your options are number A tRNA binding, number B translocation, number C peptide chain transfer, and number D incoming amino acid. Aminoglycosides work by blocking which of the following? Number A tRNA binding, number B translocation, number C peptide chain transfer, and number D incoming amino acid. Aminoglycosides work by blocking which of the following during translation? Let me know A, B, C or D, which of the following you think is the correct answer for problem number 18. Whatever you think, let me know. A, B, C or D, which of the following will be correct? Number A tRNA binding, number B translocation, number C peptide chain transfer, and number D incoming amino acid. Which of the following is blocked by aminoglycoside class of antibacterial agents? Again, I will wait for a minute. Aminoglycosides work by blocking which of the following during the translation? tRNA binding, translocation, peptide chain transfer, or incoming amino acid? Let me check the chat box. Let me check if there is any answer for problem number 18. Okay, I got one answer which is saying number B. Let me check if number B let me check if number B is the correct answer for our problem number 18. So aminoglycosides work by blocking, yes, number B, translocation during the translation or protein synthesis. So stages at which the antibacterial agent inhibit the translation process. So first we know there is a 50S and 30S ribosome, mRNAs bind with the 30S ribosome. So oxalirenone, they bind with the 50S subunit. We have discussed it earlier, right? Then in this is the translational complex. Tetracycline blocks the tRNA binding. Okay, so tetracyclic class of antibiotic, they blocks the tRNA binding. So amino acid is brought by tRNA, and this tRNA binding is blocked by tetracycline. Then we have chloramphenicol, which blocked the peptide chain transfer. So there is a growing peptide chain, and chloramphenicol, it blocks the peptide chain transfer. Then we have the macrolides, which block the translocation along with the aminoglycosides. These both of these agents, they actually block this translocation. Okay, by binding. So as you can see, this is macrolides, they block the translocation along with the aminoglycosides. Okay, so these are the various antibacterial agents: oxalirenone, chloramphenicol, tetracycline, macrolide, and aminoglycosides. All these inhibit the bacterial protein synthesis, but the method of mechanism of actions are different. Oxalirenone bind to the 50th subunit, tetracycline blocks tRNA binding, chloramphenicol blocks peptide chain transfer, macrolides and aminoglycosides, they block the translocation. Okay. Now I will go to the next problem. Problem number 19 says, which of the following is the correct reason for the drug resistance of kanamycin? Your options are: number A, elimination of the hydroxyl group; number B, phosphorylation of hydroxyl group; number C, efflux pump; and number D, target overproduction. Which of the following is the correct reason for the drug resistance by kanamycin? Elimination of the hydroxyl group, phosphorylation of hydroxyl group, efflux pump, or target overproduction? Let me know A, B, C or D, which of the following will be the correct answer. Which of the following is the correct reason for the drug resistance of kanamycin? Number A, elimination of hydroxyl group; number B, phosphorylation of hydroxyl group; number C, efflux pump; and number D, target overproduction. Again, I will wait for a minute, then I will check the chat box. The correct reason for the drug resistance of kanamycin, which will be the correct answer? Elimination of the hydroxyl group, phosphorylation of hydroxyl group, efflux pump, or target overproduction? Let me check the chat box because we don't have much time and we have another question left which we have to discuss. Okay, I got one answer which is saying that number D is the correct answer. Let me check if number D is the correct answer or not. The correct answer is number B. Phosphorylation of the hydroxyl group is the reason for the drug resistant of the kanamycin. Okay, so this is kanamycin. As you can see, there is a hydroxyl group here which is phosphorylated and which is making this particular drug inactive. Okay, this phosphorylation. So another potential approach to countering resistance is to modify antibiotics such that they gain resistance in the mechanism of resistant use against them. For example, kanamycin is an aminoglycoside which is no longer in use because the resistant bacteria can phosphorylate one of the hydroxyl group present. An active analog has been synthesized which replaces the susceptible alcohol by ketone. Okay, so if the ketone analog is substituted in kanamycin in place of hydroxyl group, what will happen? This ketone is in equilibrium with the hydrated gem diol. Okay, this ketone is in equilibrium with the hydrated gem diol, and when phosphorylation occurred to the diol, the phosphate group thus formed act as a good living group and the ketone is regenerated. This in vitro tests show that this agent was active against strains of bacteria which were resistant to kanamycin. So this is the analog of kanamycin which is active because first it is in equilibrium with this diol, and this diol can the phosphorylation of diol generates a good phosphate leaving group and which can regenerate this ketone group. Okay, so this is how this resistance can be encountered by slightly modifying the structure of a compound. So remember, kanamycin is an aminoglycoside where the phosphorylation is the reason of drug resistance, and converting the hydroxyl group to ketone is the how we can encounter the resistance. Okay. Now the last question of today's session: which of the following mechanisms is followed for the resistance of the sulfur based antibiotic? Okay, your options are: number A, target mimicry; number B, efflux pump; number C, drug inactivation; number D, target overproduction. Which of the following mechanisms is followed for the resistance of sulfur based antibiotics? Number A, target mimicry. Number B, efflux pump. Number C, drug inactivation. Number D, target overproduction. Let me know A, B, C or D, which of the following is the correct answer for problem number 20. Which of the following mechanisms is followed for the resistance of sulfur-based antibiotics? Target mimicry, efflux pump, drug inactivation or target overproduction? A, B, C or D? What will be the correct answer? Let me check the chat box. Okay, because we don't have much time and I have to discuss this question. I hope to find an answer. Okay, I got an answer which is saying number B is the correct answer. Let me check if number B is the correct answer or not. So the correct answer will be, it is number D, target overproduction. Okay, so overproduction of a substrate or ligand, so overproduction of a substrate or of a target enzyme would competitively block the ability of drug to bind at the active site. An example of the overproduction of para-aminobenzoic acid or PABA is the mechanism of resistance of the antibacterial sulfur drug. So we all know sulfur drugs are basically the competitive inhibitor. Okay, so they act as a competitive substrate for the para-aminobenzoic acid, which is the normal substrate. So if there is a target overproduction, means if there is a lot of the para-aminobenzoic acid, so what will happen? The para-aminobenzoic acid will replace the sulfur drug. So this is the mechanism of resistance of the sulfur drug, the target overproduction. Okay, so if there is, so this is a very basic thing which we have discussed when we were discussing about the competitive inhibitors. Okay, how to counter the competitive inhibition, that is that you have to increase the amount of the substrate. So this was the last question of today's session. So today basically we have discussed about the various antibacterial agents. We have discussed about the mechanism of actions, their examples, and also we have seen their structure. So this is my email ID. If you have any doubt or any query, you can let me know via email. Okay, also you can let me know via the discussion forum of the this course, Organic Chemistry and Biology and Drug Development. So today it was our week 10 live session. We will meet in our next live session, which will be on next Friday from 6 p.m. where we will discuss about the week 11 course content. Okay, so if you have any doubt or any query, please feel free to reach me. Thank you all for joining. Thank you all for answering diligently throughout the class. Okay, I am closing the session for today. Thank you all. Next, let us meet on the next Friday. Okay, thank you.